Hi everyone. In today's video, I wanted to go through the process of DNA replication. Now, what is DNA replication? Well, in simple terms, it's essentially the parent strand, which is the older strand of DNA, being copied into two new daughter strands, which contain both parts of the older parent strand and also new nucleotide bases. Therefore, we say that the process of DNA replication actually follows the semi-conservative model. Now, the semi-conservative model basically states that if we have a particular parent strand as so, and it contains two strands like this, then the two daughter strands that form due to the process of DNA replication will contain one old strand from the parent each, and will also contain one new strand of their own. So the strand denoted in green on the left daughter DNA strand, and the one denoted in crimson on the right is actually the new strands that exist, whilst as you saw in the parent strand, the blue and sort of the very light yellow color are also present within the daughter strands. So how did this process actually occur? Well, say we have a particular parent strand present here, and in this case, for simplicity, I'll only draw it in one color. Say we have a particular parent DNA strand here. There are actually a particular set of nucleotide bases, which we call the origin of replication. Now, these nucleotide bases are what code for a special enzyme called DNA helicase to allow it to come to this particular part of the DNA, bind to it, and therefore allow for the DNA chain to simply be unwound. So therefore, this part of the DNA sequence becomes separated. This allows for, and now I'll only draw it in a simplified manner. So if we have the two helices of the DNA present here, there is essentially an opening that is created as so. And this opening is maintained by small molecules, which we call single-stranded binding proteins, which I'll simply write as SSBP. These single-stranded binding proteins hold the two DNA strands apart. Furthermore, there is another enzyme called DNA gyrase. This enzyme actually allows for the DNA sequence to be cut into small pieces. This is because if you imagine DNA to be a one long rope, then twisting it in so many manners will eventually mean that there are certain kinks and unwanted sections of the DNA strand to be created. To ensure that this doesn't happen, it essentially allows for the cutting of the DNA strand into smaller sections to therefore introduce nicks and therefore allow for the DNA strand to not be arranged or not tangled in any manner. So, now the DNA strands have been separated from one another. There are special names for the different parts of the DNA strand that actually exist. Firstly, this section, which is essentially where the two prongs of the DNA separate, is called the origin of replication, but the structure as a whole is called the replication fork. Now, this replication fork is where the two strands actually separate, and the two strands that separate are called the leading strand and lagging strand. And they are called so as they have different functions. So if I were to redraw just a little bit bigger here, so the leading strand is called so as it actually follows a 5' to 3' direction. And you may be wondering, why is this important? Well, essentially, there is an enzyme called DNA polymerase 3. And this enzyme essentially is responsible for the formation of a second DNA strand that is complementary to the leading strand, and therefore allows for the formation of the daughter strand. So if I was to denote the DNA strand that is formed by DNA polymerase 3 in this color, it essentially allows for the formation of this strand. But it can only work in a 5' to 3' direction, which is why the leading strand is so important. Just to not have any confusion, I'll just draw a border here. So this particular strand is the leading strand. However, there's another hurdle that is in our way here. DNA polymerase 3 can't actually start the process of DNA replication by itself, which is why we have a special enzyme called RNA primase. Now, if we zoom in to this leading strand and redraw it here, with its various nucleotide bases present as these square-shaped structures, what RNA primase basically does is that it adds a complementary RNA sequence for about 10 nucleotides to this particular DNA leading strand. And the addition of this RNA primer, what we call, allows for the DNA polymerase 3 enzyme to bind to the leading strand. So imagine this rectangular shape is DNA polymerase 3. And this is what is able to allow for the process of DNA replication to occur, as now the DNA polymerase 3 enzyme has bound to the lagging strand. It is able to travel in a 5' to 3' direction. So, now that the RNA primer has been added, the addition of DNA nucleotides can occur, as DNA polymerase 3 has received its starting sequence. So, what happens after all of this has occurred? Say that all the nucleotide bases on the leading strand have now been added. Well, there's a special process that occurs. Obviously, as the RNA primer, which was present on these 3 different nucleotides, is still here, we need to remove it because we can't have a RNA sequence in our new DNA sequence. So there's a special enzyme called DNA polymerase 1. And this enzyme is able to replace this RNA sequence with an appropriate DNA sequence. So essentially, the new RNA strand is now complete. However, we still have another problem present here, as even though the RNA strand may be replaced, there's still some sort of a break present here, because essentially we've replaced a new DNA sequence here next to the pre-existing DNA sequence that DNA polymerase 3 added. So as these two sequences do not match with one another, there's another special enzyme called DNA ligase. And this DNA ligase is able to allow for the fixing of various breakages in the phosphodiester bonds or the sugar phosphate backbone that exists between the nucleotides in order to ensure that the new DNA sequence is as tough and strong as possible. So, this eventually means that after all of this has occurred, the process of DNA replication on the leading strand is complete. However, the process is not as simple on the lagging strand. As I mentioned before, as the leading strand is located or oriented in the perfect position of being 5' to 3', the DNA polymerase 3 enzyme is able to just coast through in this direction. However, the lagging strand, as its present anti-parallel, is actually present in a 3' to 5' direction. So what does this mean? Well, essentially, it means that the DNA polymerase molecule has to work in particular segments in order to allow for the process of DNA replication to occur. And these segments are referred to as Okazaki fragments. As the direction of the DNA polymerase enzyme essentially does not agree with the direction of the DNA chain, what happens is that the DNA polymerase enzyme works in particular segments from a 5' to 3' direction in order to allow for DNA replication to occur. These segments are called Okazaki fragments, named after the scientists that discovered them. Now, if we zoom in a little closer to this strand, we will actually see that the nucleotide bases first follow the same process, in that for about 10 nucleotides, an RNA primer is added. Then, the DNA polymerase enzyme binds and works in a 5' to 3' direction. However, say for example, the DNA polymerase enzyme has already worked on this section of DNA, and the new section it starts working on is starting from here. Well, what happens in that case is that the DNA polymerase 3 enzyme moves towards this direction and stops when it reaches here. This essentially means that as the DNA polymerase enzyme adds its nucleotide bases, first, the DNA polymerase 1 enzyme must replace this RNA primer with an appropriate DNA sequence. After that has been done, DNA ligase must attach the appropriate bond here in order to allow for no breakages in the sugar phosphate backbone. And finally, another bond must be added by the DNA ligase enzyme between these two fragments in order to allow for another repairing in the break between the sugar phosphate bond. So that's essentially how DNA replication continues. And as you can see, it is a highly complicated process involving many enzymes. Now, for good reason, it's considered a highly complicated process, as it is also an important process. So, because it's so important, we need to ensure that there are no mistakes in the actual replication process. The absence of, or if there are mistakes present, the fixing of these mistakes is ensured by an activity called DNA exonuclease activity. Now, DNA exonuclease activity is actually quite important, as DNA replication on average has an error every 1 in 1 billion nucleotide bases. Now, you may be wondering, 1 in 1 billion, that's a pretty small chance of a mistake happening. However, when you take into consideration that we have trillions of cells in our body, and those cells within them have trillions of nucleotide bases, you can see that this is a pretty high chance. So what DNA exonuclease activity does is it basically allows for the proofreading of the DNA strand. So imagine it in the way you're writing a book. Essentially, the DNA exonuclease activity will be the publisher of that publishing company or any particular person that is involved in the checking of that book, ensuring that you have no spelling mistakes, as essentially it's the same thing. The spelling of the DNA sequence is dictated by these 4 nucleotide bases. So the exonuclease activity has to check that the C should not be in the position of the T, or a G must not be in the position of an A. This proofreading is coupled with actually fixing the mistake. So the exonuclease activity is able to be assisted actually also by DNA polymerase 3 in order to allow for the fixing of these particular mistakes. So as you can see that there are a lot of different ways in order to allow for this process to go well, and there are a lot of safety nets that ensure that this process doesn't go awry. So a quick review. DNA replication is the formation of 2 daughter strands from one parent strand using the semi-conservative model. It involves a DNA helicase enzyme binding to the DNA chain in order to allow for its separation. Then, it also allows for single-stranded binding proteins to hold this apart. A DNA gyrase enzyme which introduces NICs, which allows for less tension in the DNA strand. There is a leading and lagging strand. The leading strand can be replicated in a 5' to 3' direction. The lagging strand, on the other hand, must be done in a 5' to 3' direction, but away from the origin of replication, therefore meaning that there are formation of Okazaki fragments. And the actual process involves an RNA primase enzyme adding an RNA primer to the nucleotide base first, then the DNA polymerase 3 enzyme replacing this with the appropriate, or starting its DNA replication process with the appropriate DNA nucleotide bases in a 5' to 3' direction. After this has occurred, DNA polymerase 1 is able to replace the RNA primer with an appropriate DNA sequence. And finally, DNA ligase is able to replace any kinks or breakages that may have occurred in the sugar-phosphate bond. After all of this, DNA exonuclease activity, even though DNA has a success rate of mistakes of only 1 in 1 billion, is able to allow for extra proofreading and fixing of any different mistakes that may occur with the assistance of DNA's polymerase III. So, this is the process of DNA replication.